Hello and welcome back to lecture 9.1, where we will be talking about cellular respiration. We're going to do an introduction, and then we're going to go through the first two steps in cellular respiration, glycolysis and pyruvate oxidation. Great. Before we get to that, though, let's, here we go. Which of the following are true when comparing and contrasting light-dependent reactions and the Calvin cycle? So this was lecture question 8.3, obviously. And if you said B, you'd be correct. Great. Moving on. Here you go. Lecture 9.1. Actually, before we get started, you might want to do something like this ahead of time. So you can probably come up with a better table in the long run, but this is not a bad way to start kind of organizing yourself a little bit here. And keeping track of all the different products of these different processes and maybe their products. You have different steps that undergo different parts that undergo different processes that end up with particular products that happen to be the parts that start the next process. Anyway, so here it is, cellular respiration, which is the process that most eukaryotes use to convert carbohydrates in the form of glucose into a more usable form of energy. This is the overall equation with a glucose molecule and 6 molecules of oxygen gas as the major reactants, and 6 carbon dioxides, and 6 molecules of water, and energy, of course, as the products of the reaction. Now, key to this whole process is kind of following the hydrogens and keeping an eye on what is being oxidized and what is being reduced. So based on what we can see here, what do you guys think is being oxidized in this process? Right, the glucose is being oxidized. So it's being oxidized from glucose into carbon dioxide, right? Now, what are the things that are being reduced then? Yeah, oxygen. Oxygen is being reduced into water. Okay, so the most efficient way, or in order to make this happen the most efficient way possible, what we're going to be doing is kind of using some intermediates to help shepherd along these electrons from the sugars until we pass it off to the oxygen. Any guesses as to what kind of molecules we'll be using to do the shepherding? Yeah, that's right. So NAD plus and FAD are two examples of this, and they're both electron carriers, something you should be pretty familiar with at this point, right? We talked about that a lot during photosynthesis, that lecture you just watched. Okay, so they're going to basically, how these work, these electron carriers like NAD plus and FAD, they're going to come up to that glucose molecule, or what's left of it, depending on when they get there, and strip off some hydrogen, really some electrons and maybe some hydrogen ions as well. Then they're going to pass off these electrons and possibly the hydrogens to later reactions. Okay, specifically, they're mostly going to be using these electrons to fuel an electron transport chain, but we'll get to that later on. Now, if you notice, NAD plus actually manages to pull off more electrons than hydrogen ions, unlike FAD, which actually pulls off an equal number of hydrogen ions and electrons. Now, both FADH and FADH2, let's say the reduced versions, I had to do reduction is gain in my head, the reduced versions of NAD plus and FAD, they basically represent this kind of stored energy that we're going to harness later on. Okay. Speaking of stored energy, remember ATP? Do you remember how it stores energy? Where do we get all this energy that we can shepherd around with ATP? Right, that third phosphate group here, this bond between the second and third phosphate. So ultimately, what cellular respiration does is recreate that bond or phosphorylate an adenosine diphosphate. So that's kind of our ultimate goal of this whole process, is reattaching a phosphate onto an adenosine diphosphate and recreating that spring-loaded bond. So, how do we think it gets there? Well, actually, that's kind of the whole rest of the lecture. So stay tuned, and you'll have to probably watch all lecture 8.2 as well to figure out what's going on exactly. Anyway, well, we can do a brief overview at least of cellular respiration before diving it in, kind of give yourself a bit of a groundwork here. So there are 4 steps to this process. There's glycolysis, number 1. There's oxidation of pyruvate to acetyl-CoA. Bit of a mouthful there. There's also the citric acid cycle. And there's oxidative phosphorylation. It's the final step. Okay. Now, for each of these stages, what you're going to want to understand about it is, so you're going to understand why it's occurring or the purpose. You're going to understand want to understand the place where it's happening in the cell or mitochondria. You're going to want to know the parts that kind of enter each stage, or the molecules that enter each stage, and then what happens to them. In other words, the process those parts undergo. And then finally, you're of course going to want to know the products of the processes, right? So the products of the process the parts undergo. Keep saying something. Why is this striking something familiar? Products, parts, process. I don't know. Anyway, so more or less, we're going to want to keep track of also kind of some specific things that are getting done in this process. We're going to want to keep track of how many carbon dioxides get produced. We'll also, of course, definitely want to know how much ATP is made in this process, or in each step of this process. That's kind of the whole point of this after all, right? We'll also want to keep an eye on how much NADH and how much FADH2 are made, right? So we're going to try to keep a kind of running tally of that on the top right here of our screen. So just keep an eye on that.